Oxidation of fatty acids. Fatty acid breakdown can be divided into a three-part process. First, the fatty acid must be activated by bonding to coenzyme A. And then the fatty acid must be transported into the mitochondrial matrix by a shuttle mechanism. The fatty acid must be repeatedly oxidized, cycling through a series of four reactions to produce acetyl-CoA, FADH2, and NADH. Fatty acid activation. This is the first stage of fatty acid oxidation, and it takes place in the outer mitochondrial membrane. In here, the fatty acid is converted to a high-energy derivative of coenzyme A. This requires one ATP molecule wherein two high-energy phosphate bonds are broken down, so the ATP is converted to AMP. The activated fatty acid CoA molecule is called an acyl-CoA. Now, what is the difference between an acyl-CoA and an acetyl-CoA? Acyl refers to a random length fatty acid carbon chain, so as represented by R, which refers to any carbon chain of any length, while acetyl is specific and it refers to a two-carbon chain covalently bonded to coenzyme A. Since acyl-CoA is a large molecule, it cannot pass through the inner mitochondrial membrane to the mitochondrial matrix where the enzymes needed for fatty acid oxidation are located. The inner mitochondrial system has a group-specific transport system for fatty acids. So the acyl groups of the activated fatty acids are transferred to a free carnitine by an enzyme called carnitine acyltransferase. So here, the free carnitine attaches with the acyl group and then it enters the matrix as acylcarnitine. Once inside, the mitochondrial enzyme carnitine acyltransferase catalyzes the return transfer of the acyl residue to coenzyme A. So the carnitine shuttle is the rate-determining step in mitochondrial fatty acid degradation. Okay, how about the beta oxidation pathway? So the beta oxidation pathway can be defined as a repetitive series of four biochemical reactions that degrades acyl-CoA to acetyl-CoA by removing two carbon atoms at a time, with FADH2 and NADH also being produced. For a saturated fatty acid, the beta oxidation pathway involves the following functional group changes at the beta carbon and the following reaction types. So each repetition of the four reaction sequence generates an acetyl-CoA molecule and an acyl-CoA molecule that has two fewer carbon atoms. So here you will notice that we started with an alkane, and then because of the hydrogenation, a double bond is formed, so we therefore have an alkene. And upon the addition of water or by hydration reaction, a secondary alcohol is produced. And then a second dehydrogenation reaction takes place and a ketone is produced. And then thiolysis or chain cleavage to produce an acetyl-CoA molecule and an acyl-CoA molecule that will again undergo the four biochemical reaction. So here, in the following slides, we are going to discuss in detail the steps of the beta oxidation pathway. First step is the first dehydration. So in this step, hydrogen atoms are removed from the alpha and the beta carbons. And because of their removal, a double bond will be formed between these two carbon atoms. FAD acts as the oxidizing agent and an FADH2 molecule is the product. The enzyme involved here, the acyl-CoA dehydrogenase, is stereospecific in the sense that only trans double bonds are produced. Step number 2 involves hydration, so that means it involves the addition of water. So here, a molecule of water is added across the trans double bond, producing a secondary alcohol at the beta carbon position. And then for the third step, this is the second dehydrogenation reaction and also an oxidation reaction. So here, two hydrogen atoms are again removed, converting the beta hydroxy group to a keto group. NAD plus serves as the oxidizing agent, and the required enzyme exhibits absolute stereospecificity for the L isomer. In the fourth step, the fatty acid carbon chain is broken between the alpha and beta carbons by reaction with a coenzyme A molecule. The result is an acetyl-CoA molecule and a new acyl-CoA molecule. However, this one is shorter by 2 carbon atoms than its predecessor because the two carbon atoms removed are this one in the acetyl-CoA. The new acyl-CoA molecule, now with shorter 2 carbons or now shorter by 2 carbons, is recycled through the same set of 4 reactions again. So this will yield another acetyl-CoA. And then recycling takes place again and again until the entire fatty acid is converted to acetyl-CoA. So the fatty acid carbon chain is sequentially degraded 2 carbons at a time. The chain cleavage reaction that takes place in this step is called thiolysis by analogy with the process of hydrolysis, which also involves the breaking of a molecule into 2 parts. So it is the thiol group of coenzyme A that undergoes the reaction. Here in the fourth step of the beta oxidation of the saturated fatty acid, for example, stearic acid with 18 carbons, you will see here the many loops in the diagram. Each loop of the pathway represents a repetition of steps 1 to 4. The number of acetyl-CoA produced from the beta oxidation pathway is actually equal to half the number of carbon atoms in the fatty acid. So that means for stearic acid having 18 carbon atoms, the number of acetyl-CoA that will be produced are 9 molecules of acetyl-CoA. And also, for example, for a fatty acid containing 14 carbon, 7 acetyl-CoA molecules will be produced. The number of repetitions of the beta oxidation pathway that are needed to produce this acetyl-CoA is always 1 less than the number of acetyl-CoA molecules produced because the last repetition produces 2 acetyl-CoA molecules as a carbon 4 unit splits into carbon 2 units. So as you can see in here, for stearic acid having 18 carbons, a total of 9 acetyl-CoA will be produced, and it takes 8 repetitive sequences for this acetyl-CoA to be produced. So for a 14 carbon fatty acid, 7 acetyl-CoA will be produced, and it takes 6 repetitive sequences for this acetyl-CoA molecules to be produced. Now let us discuss the beta oxidation of unsaturated fatty acids. The dietary lipids that we consume is not only composed of purely saturated fatty acids. Of course, unsaturated fatty acids are also common components. However, their oxidation requires 2 additional enzymes besides those needed for oxidation of saturated fatty acids. The 2 enzymes are an epimerase that can change a D configuration to an L configuration, and a cis-trans isomerase that causes the production of a trans 2,3 double bond from a cis double bond. So this one is an important step so that step number 2 in the beta oxidation can proceed in the normal fashion. ATP production from fatty acid oxidation. This shows the number of ATP that will be produced from one molecule of stearic acid, a fatty acid with 18 carbons. From the previous slides, we learned that a stearic acid produces 9-acetyl-CoA through fatty acid oxidation, which requires 8 repetitions. For each 4-reaction sequence or repetition, there are one FADH2 molecule and one NADH molecule that are being produced, which totals to 8 molecules of each since there are 8 repetitions. So for every stearic acid molecule, the number of ATP produced is 122. Here, we are going to compare the number of ATPs produced from a complete oxidation of glucose and stearic acid. For every one glucose molecule, a total of 30 ATPs are produced, while for one stearic acid, an estimate of 120 ATPs are produced. Well, we know that glucose has 6 carbons and stearic acid has 18 carbons. How about if we compare glucose and stearic acid on the basis of the same number of carbons? So here, we have 3 glucose molecules amounting to a total of 18 carbons and 1 molecule of stearic acid. Here we can see that still, 1 molecule of stearic acid produces more ATP. On the basis of equal numbers of carbon atoms, lipids are 33% more efficient than carbohydrates as energy storage systems. How about if we compare glucose and stearic acid on a mass basis? Here we can see that on an equal mass basis, so for example, we have 1 gram of stearic acid and 1 gram of glucose, fatty acids produce 2.5 times as much energy per gram as carbohydrates. So which is the preferred fuel of the body? Is it glucose or is it fatty acids? Knowing that fatty acids yield 2.5 times as much energy per gram as glucose, is it fatty acids? So in a normal functioning human body, certain organs use both fuels, glucose and fatty acids. Others prefer glucose and still others prefer fatty acids. Some of the generalization about fuel use are: first, skeletal muscles use glucose from glycogen when in an active state. So for example, when our muscles contract, okay, it prefers glucose over fatty acids. In a resting state, skeletal muscles use fatty acids. Cardiac muscles depend first on fatty acids and secondarily on ketone bodies, glucose, and lactate. The liver uses fatty acids as the preferred fuel. And lastly, brain function is maintained by glucose and ketone bodies. So that's it, and see you in our next topic.